Introduction to Recombinant DNA or DNA technology. Recombinant DNA technology is a revolutionary advancement in genetic engineering that involves combining genetic material from different sources to create new DNA sequences with specific functions. These techniques enable scientists to manipulate an organism's genetic makeup, introducing desirable traits that enhance its characteristics or capabilities. Since its developed in 1970s, our DNA technology has transformed multiple fields, including medicine, agriculture, industry, leading to groundbreaking applications that benefit society in numerous ways. So, how our DNA technology really works? The process of creating recombinant DNA involves several key steps. So, step number one, isolation of the desired gene. Scientists identify and isolate a specific gene that carries a beneficial trait. This gene is often obtained from a donor organism, such as bacterium, plant, or animal. For the second step is the insertion into a vector. The isolated gene is inserted into a carrier molecule known as a vector, often a plasmid or a virus, which facilitates its transfer into the target organism. For number three, transformation of host cells. The recombinant DNA is introduced into a host cell such as bacteria, yeast, or plant cells where it integrates with the host genome or functions independently to express the desired trait. And for the last step, replication and expression. Once inside the host, the recombinant DNA replicates and produces the intended protein or characteristics, which can be harnessed for various applications. So, by using this technique, scientists can create genetically modified organisms with enhanced properties, ranging from disease resistance in crops to production of therapeutic proteins in bacteria. Now let's talk about the applications of rDNA technology. rDNA technology has significantly impacted modern medicine by enabling the production of life-saving drugs, vaccines, and gene therapies. Notable contributions include production of biopharmaceuticals. Recombinant DNA allows for the mass production of the essential medication such as insulin for diabetes treatment, human growth hormone, and clotting factors for hemophilia patients. It also includes vaccine development. Our DNA technology is used to develop vaccines, such as the recombinant hepatitis B vaccine, which stimulates the immune system without using live pathogens. And lastly, gene therapy. Scientists use recombinant DNA to treat genetic disorders by correcting faulty genes. Conditions like cystic fibrosis, sickle cell anemia, and muscular dystrophy are being targeted for potential cures through gene editing techniques like CRISPR.
Agricultural improvements. In agriculture, rDNA technology has led to the creation of genetically modified crops that offer numerous benefits including pest and disease resistance. Crops like BT corn and BT cotton have been engineered to produce insecticidal proteins reducing the need for chemical pesticides. Herbicide tolerance crops such as glyphosate resistant soybeans allow farmers to control weed more effectively, leading to increased yields. Enhanced nutrition. Biofortified crops like golden rice enriched with vitamin A aim to combat malnutrition in developing countries. 3. Industrial environmental applications. Beyond healthcare and agriculture, rDNA technology has revolutionized industries and environmental conservation. Biodegradable plastics and biofuels, genetically engineered microorganisms are used to produce sustainable alternatives to petroleum based products. Bioremediation. bacteria modified through rDNA technology can help clean up oil spills and remove toxic pollutants from the environment. Enzyme production. Recombinant enzymes are widely used in industries such as food processing, detergent manufacturing, and biofuel production. Ethical and regulatory considerations. While rDNA technology offers numerous benefits, it also raises ethical and safety concern issues such as the long-term effects of GMOs, genetic privacy in gene therapy, and potential ecological risk require careful oversight. Governments and scientific organizations have established regulatory frameworks to ensure that rDNA applications are safe and ethically responsible. In conclusion, recombinant DNA technology has revolutionized science and industry, offering solutions to critical challenges in medicine, agriculture, and environmental sustainability. By enabling precise genetic modification, this technology continues to drive innovation, improving human health, food security, and industrial processes. As research advances, responsible application and regulation will be essential to maximize its benefits while addressing ethical and safety concerns.
Recombinant DNA. Technology is groundbreaking scientific advancement that has revolutionized the field of medicine, particularly in the production of pharmaceuticals and development of innovation treatment for genetic disorder. This technology involves the manipulation and recombinant of genetic material from different sources to create modified DNA. Sequence with desired traits which can then be introduced into the host cells to produce therapeutic proteins, hormones, and other biologically active substances. One of the most significant applications of rDNA technology is the production of insulin, a hormone. Agriculture. Genetically modified crops are developed using recombinant DNA technology to introduce specific traits that enhance their performance, making them more resilient and beneficial for farmers and consumers. This process involves altering the genetic makeup of a plant by inserting genes from their organisms, such as bacteria or other plants, to achieve desirable characteristics. One of the primary reasons for developing GM crops is to improve yield by making plants more resistant to environmental stresses, pests, and diseases. For instance, Piti cotton has been genetically engineered to express a protein derived from Bacillus thuringiensis, a naturally occurring bacterium.
Economic impact. The commercialization of genetically engineered products has led to significant economic benefits. Pharmaceutical companies profit from biotech drugs, which farmers experience increased crop yields and reduced losses due to pest-resistant GM crops, lowering overall production costs. So, based on my knowledge, commercialization of genetically engineered products has greatly impacted the economy. Pharmaceutical companies benefit from selling biotech drugs while farmers gain the higher crops, yields, and fewer losses thanks to pest-resistant GM crops. So this reduces production costs, making farming more efficient and profitable. Overall, these advancements contribute to economic growth by improving productivity and lowering expenses.
Environmental impact. rDNA technology contributes to environmental sustainability by reducing the need for chemical, pesticides, and fertilizers. However, concerns remain regarding potential risks, such as unintended effect on biodiversity and the emergence of resistant pests.
and health concerns. The safety of genetically modified organisms, or known as GMOs, remains a central topic of ethical debate, particularly regarding to their potential effects on human health. While regulating agencies such as the US or the Food and Drug Administration, the European Food Safety Authority, and the World Health Organization conduct a rigorous safety assessment before approving by the GMOs for commercial use. Concerns persist about their long-term impact, potential health risk, and allergenicity. One of the main concerns surrounding the GMOs is their potential to introduce new allergens or exacerbate existing allergies. When genes from the different organism are inserted into crops or food product, there is a possibility that the introduced proteins could trigger unexpected allergen reactions.
Future prospects of recombinant DNA technology. Recombinant DNA, rDNA technology, has transformed numerous scientific fields, including medicine, agriculture, and environmental science. Its ability to modify genetic material has led to groundbreaking advancements, from life-saving gene therapies to genetically engineered crops that enhance food security. As the technology continues to evolve, its potential applications will expand, addressing global challenges such as disease treatment, climate change, and sustainable resource management. However, the widespread adoption of rDNA technology must be accompanied by responsible regulatory measures and ethical considerations to ensure its safe and equitable use.